Let's discuss neuropathology. There are several types of cells in the brain that are worth reviewing. Neurons are the primary information transmitting cells in the CNS. Glia are important supporting cells that help to maintain neurons. Astrocytes, oligodendrocytes, and ependymal cells are the major types of glial cells. Microglia are not truly glia per se, but are actually immune cells from the macrophage lineage. Choroid, Plexus, epithelial cells are a type of epithelium in the ventricles that make CSF. Astrocytes are important glial cells with a number of different functions. They are important in supporting the structure of the normal brain and the repair response to injury, including formation of glial scars. Astrocytes contribute to the blood-brain barrier formation. They insulate synaptic clefts between an axon and dendrite and prevent excess neurotransmitters from leaking. Astrocytes also regulate extracellular potassium levels around neurons so that neurons can properly fire action potentials. Here's an example of an inactive astrocyte on H&E stain. Astrocytes are smaller than the cell bodies of neurons. On H&E, they have an irregular oval shape and have a lighter nucleus than oligodendrocytes. Staining for glial fibrillary acidic protein, or GFAP, can help to highlight astrocytes and differentiate inactive astrocytes from active astrocytes. On the left, you can see the rami of inactive astrocytes, including processes that help form the wall around a capillary. The right shows active astrocytes with the gorments of the cell body. Oligodendrocytes send up processes that wrap around axons to form a myelin sheath. Unlike Schwann cells in the peripheral nervous system, a single oligodendrocyte will myelate multiple axons. Oligodendrocytes are best seen on periodic acid Schiff, or PAS, stains. They have small, round, hyperchromatic nuclei. Here's an electron micrograph of an oligodendrocyte wrapping around an axon in the CNS. You can see that the oligodendrocyte wraps around the axon multiple times. The choroid plexus epithelium is made up of cuboidal cells, mostly located in the lateral ventricles and fourth ventricle. However, they're also present to some degree in the third ventricle. These cells produce the CSF that flows through the ventricles. Ependymal cells line the ventricles. These help to direct the flow of CSF. Here's an H&E stain of the choroid plexus. You can see that it consists of cuboidal epithelial cells surrounding vessels. Fluid from the blood passes through the choroid epithelial cells and enters the ventricles as CSF. Here's an H&E of ependymal cells lining the ventricles. You can see cilia extending from them into the ventricles. Interestingly, functioning cilia do not seem to be necessary to have normal CSF flow. Dysfunction of the cilia, such as in primary ciliary dyskinesia, does not produce hydrocephalus in humans. Microglia are the resident immune cells of the CNS. They are distributed throughout the brain in order to be ready to respond to pathogens or to structural brain damage. When they become active, they can take on two different morphologies depending on the underlying pathology. In viral infections, they form rod cells, which surround virally infected neurons and form a microglial nodule. In structural problems, like in tissue infarction, they become foamy macrophages, which can remove broken down tissue. Here's a special LCA stain showing inactive microglia acting as sentries for the brain. Here are microglial rod cells active in a viral infection, and here are foamy macrophages removing necrotic tissue. Gray matter is where neuron cell bodies are located in the CNS. Gray matter is also where synapses are located in the brain, so axon terminals and dendrites of neurons can also be found in the gray matter. The white matter is mostly made up of projecting axon tracts traveling to other locations in the brain, with myelin made up of oligodendrocytes. However, oligodendrocytes are also found in the gray matter because the axons still travel through the gray matter before reaching the location where the synapse on dendrites. Astrocytes can be seen in both gray and white matter. Microglia are distributed evenly throughout the brain in both white and gray matter, but are hard to see on H&E in brains without disease. The cerebral cortex and basal ganglia and thalamus, not pictured here, are made up of gray matter. Here is the white matter, located deep to the cerebral cortex. You can also see white matter running through the basal ganglia as the internal capsule. This is gray matter on H&E stain. You can see large neuronal cell bodies, dark oligodendrocytes, and oval astrocytes. All of this eosinophilic material between the cells is called neuropil, is where axons and dendrites run. Here's white matter on H&E stain. You can see many oligodendrocytes present myelinating the axons running through the white matter. Let's talk about a few pathologic features of the CNS. Early after infarction, in about 12 to 24 hours, neurons become very eosinophilic and the nuclei become pyknotic, which means dark and shrunken. You can remember these as red dead neurons. After 24 hours, the neurons disappear as they're carted off by foamy macrophages. These foamy macrophages are differentiated from microglia and can be found inside the cavity of infarcted tissue chronically. Reactive astrocytes can be found at the edge of infarcted tissue, walling off the cavity. On the left, we have examples of normal neurons. On the right, we have several examples of newly infarcted red dead neurons. You can see that the nuclei are pyknotic and dark, and the cytoplasm is more eosinophilic. Because the brain has a very soft consistency, rapid acceleration and deceleration can cause injury. The soft brain strikes against the inside of the skull and capillaries can break, resulting in a contusion. This most commonly occurs in the areas where the brain is pressed against the skull base. So, the three most common locations are the orbitofrontal cortex, the anterior end of the temporal lobe, and the occipital pole in the back of the brain. In addition to superficial contusions, rapid acceleration and deceleration injuries can sever the axons in white matter. When an axon is severed, the cytoplasm in the neuron keeps flowing down the axon, but the cytoplasm ends up reaching the tip where the axon was severed and it can't go any further, so the tip balloons out and forms an axonal spheroid. Here are examples of axonal spheroids using different stains. When the axons of lower motor neurons are severed in peripheral nerves, the body of alpha motor neurons in the spinal cord undergoes specific changes. They undergo something called chromatolysis, which is a regenerative response that allows the alpha motor neuron to attempt regeneration of its axon. The nucleus moves to the side of the cell body, and the cytoplasm of the cell body swells. Here's an H&E of normal alpha motor neurons and an alpha motor neuron undergoing chromatolysis. The nucleus is pushed off to the side, and its cell body is swollen. We'll end with hydrocephalus. You probably remember talking about hydrocephalus during anatomy last year. There are two types of hydrocephalus, non-communicating and communicating. Non-communicating hydrocephalus occurs when there is blockage in the path of normal flow of CSF, such as at the foramen of Monroe or the cerebral aqueduct. Communicating hydrocephalus occurs when the arachnogranulations aren't able to adequately reabsorb CSF, so hydrocephalus occurs without an anatomically defined obstruction.